Hello, hello.
Hello.
Oh, hello. We're gonna get started. Thank you guys for being here. All right, so here's our, yeah, there is an echo. Oh, maybe that's,
Maybe.
maybe. Mute. I had it on mute. I'm not supposed to do that.
I think it's from someone else.
Anybody sign in on Zoom? No. Echo. New projector. Good? Okay, that sounds good. Okay, thank you, Andrew. So we're gonna get started. Here's our program for today. I'm gonna start us off with avocado industry issues. So you guys know, we got a lot of growers in here, a lot of big growers. There are some issues. So issue.
Click on the PowerPoint.
Okay, excellent. All right, that was one issue. Here's another. Phytophthora root rot. So, Romina, Dr. Gaziz is gonna go over this on laurel wilt very well. So I'm just gonna touch on this, but it is a problem. It's considered the most important and most widely distributed disease of avocado. It's reported in 70 countries and has a wide host range of over 1,000 varieties and species. So it's definitely can be a problem. In South Florida, it is everywhere, and it's a problem following excessively wet or flooded soil conditions. But we haven't seen that for a while, but we know in the past, when that's happened, it's popped up. It's caused the loss of tens of thousands of trees in South Florida since the 60s. So definitely a problem. Laurel wilt, I think you know this pretty well. So it's in 12 states. I won't get into it too much, but you can see it's all the way over to Texas. And it's getting a little lower. And we know that it's in all Florida counties. So if you're thinking of growing avocados further up north and you think that laurel wilt is not there because you don't have a lot of avocados there, it is there in the native trees. So be careful. And that's what it looks like when it gets in a grove and you don't do anything. And this is what it looks like. So we know this. And Romina will talk more about this. So what about diseases that we can control? Unlike laurel wilt, that is very difficult to suppress. We have anthracnose, we have scab, sun blotch, and not a disease, but a problem, sunburn, pests, avocado lace bug. This shows up going into the winter every year really heavily. Drip damage, avocado leaf roller, that's something that we see as well. It's on the leaves and on the fruit. Now here's one that we might not be thinking of. This is a problem, lack of fruit. We don't have enough fruit. So here is the acreage of Miami-Dade County since 2002. You see we're down over 2,000 acres. Primarily the laurel wilt and Irma. This is over 20 years. So I've been in the avocado administrative committee meetings where the big packing houses have said, we would go out of business if we couldn't use imports, because we just don't have enough fruit from Florida. Okay, so we went from, these are bushels, over 1 million, down to 400,000. We averaged 1 million, 2007 to 2011. Right now, we're negative 60%. So that is a lot. Production should begin to rise though, because there's been a lot of replanting. So because of laurel wilt, a lot of growers have put new trees in the last 5 years, so we're gonna see that production pick up. So imports, always a problem. And with the imports, the volume is so large, prices are going down. They have a lot of money for marketing, and they come from Mexico, Peru, Dominican Republic, Chile, and California. Not an import, but seasons and cultivars. We have a lot of old cultivars. You look at the list here, the picking schedule, you'll see things like 232, Pinelli, K9, Blair, Ajax, Gossman, Leona, Chica. So if you're growing those, there might be difficulty to market that. And we have a 3 to 5 month gap where we don't have the ability to put out fruit. And I know the growers are working hard to get cultivars to fill that gap. We don't have enough trees, so we talked about replanting. So unless you're a really big grower who's making their own trees, it's difficult to get enough trees to replant. And then finally, development. Every one of you has seen the development in the Redland. When you drive, it's to me, it's shocking how much there it is. This is right here where I go to work every day, 288th. They're putting in these buildings here, and all these Bismarck palms, they're all gone as well. And what you're seeing here, there it is, the Redland soil being pushed out. So that's kind of a symbol of what's going on. Okay, thank you guys. Our next speaker, who will be very brief, I'm sure, Dr. Gaziz.
Jeff, I had a question real
Yes,
quick
sir.
on the, so even though the production has gone way down with the imports, I'm guessing the prices have not changed much? Even with the drop of the production, or have prices gone up at all? Or
Down.
Yeah,
they're going down?
yeah, so not good.
Now they're going down. Summertime was good.
It's coming around.
So good morning, everyone. I don't have good news either. So I'm the pathologist for tropical fruits here at Trek. So I'm always delivering bad news, but hopefully there's some hope that you will see within this presentation on the progress we are making towards two major diseases. One is laurel wilt and the other one is phytophthora root rot. So many of you, as Jeff mentioned, know what is laurel wilt because painfully have gone through the process of removing trees and mitigating the effect of the disease. But for people that are perhaps new to the business or it's online, seeing it from different states or different countries, laurel wilt is a disease complex. The causal agent is a fungus in the Ophiostomatalis group. It's a filamentous fungus that invades the vascular system of hosts. Is a nutritional symbiont of its vector. The main vector is the red bay ambrosia beetle, a very, very tiny beetle, like 2 millimeters length beetle. And this beetle has the particularity that infests apparently healthy trees. which is not very common for ambrosia beetles or bark beetles. And so the female is the one that carries the fungus in pockets called macanger, and the female burrows and makes galleries within the tree to farm the fungus. So the beetle does not eat wood or any plant material. It feeds exclusively from the fungus that it grows. So it is a farmer, right? It farms the fungus. And so this fungus, unfortunately, is very virulent. And it's virulent because, not because it secretes toxins or kills the tissue. It's virulent because it can multiply very fast once it gets into the host. So this is previous work by Randy Plotz, and he showed that as little as 40 spores are enough to cause the symptoms and the death of mature trees. So that's very little inoculum in comparison to other diseases out there. So the female vector of this patho system, disease complex, can carry up to 6,000 spores. So that means that one female is enough to kill a tree and to start an outbreak. Because also these females have a reproductive system that they don't need to be fertilized already to start a colony. They can lay eggs and then mate with the sons and then start an outbreak. So only one female is enough to start an outbreak. The good news is that this species is not attracted to avocado. That's the good news. The bad news is that in Florida, we have a suite of other beetles that are now carrying this fungus. So this beetle is invasive to the Americas, but it came with its nutritional symbiont, which is the fungus. And once it established in the Americas, in the US, the fungus moved into other beetle systems. And now these other beetles, which are already established in our area and actually throughout Florida and throughout Central and South America, actually these are very common beetles. Now they are carrying the fungus in our state, and also it has been reported in Tennessee and other northern states as alternative vectors. So the interaction of the fungus and the host results in the occlusion of the xylem vessels. As you know, the xylem vessels are the ones that will transport the water from the base of the tree, from the roots into the canopy, right? And so unfortunately, when the beetle makes a gallery, these beetles go really deep into the trunk and they will reach the active xylem. And so when they make their gallery, they inoculate the fungus, and the fungus accidentally escapes these vessels, these galleries, into the water stream. And then it starts becoming systemic. It starts distributing itself and growing, multiplying within the vascular system. That induces defense mechanism, which is widespread in trees. That is called tylosis. Hello, sorry. Yeah. And so it will induce the production of tylosis, which is a defense mechanism, and it will block the movement of the fungus, but it also blocks the movement of the water. So that's when you get these occlusions and death of the vascular system. So this graph is showing a cross-section of a stem. As you can see, the color purple diminishes as the disease progresses. So that means that the dye stops moving through the vessels. So as the disease progresses, as the days go by from the day of inoculation, the vessels are more clogged and you cannot see the dye anymore. So that means that there is a blockage of water transport, okay? And so you can see here, clearly there is drought symptoms, right? You see first a wilting, and then a brown wilting, and then a completely dead of the plant. So a tree can die in a matter of a month, and this could be really huge, mature, producing old trees that will be killed in less than a month. that is a very lethal and aggressive fast disease. And so it has caused tremendous damage to the natural ecosystems. Again, as Jeff mentioned, if you are thinking about growing avocado up north, and you think you are okay because there are not many plantings around, think about the type of vegetation that you have surrounding these plantings. Because if you have lauraceae, which now the population is very low because this disease has killed millions of trees, but there's still some swamp bay and some red bays in the area. And so those are hosts not only for the beetle, but for the fungus as well. And so you have to think about the surrounding vegetation. And of course, it has caused a lot of damage to the avocado industry in our state. So in orchards, what happens is we have a first introduction of the fungus by any of these well-established beetle populations. And they will inoculate the tree. They will make their galleries, and in the process, they will inoculate the trees. And then if you don't remove the tree on time, the fungus colonizes the root system. And because we have large trees and our soil is so almost nothing, right? Very thin, we have the roots of the avocados like this, right? So very likely that they are connected. And we have been planting them very closely as well with large canopies. And so they are very likely to be connected. So you will see this kind of pattern of disease spread, right? That you are losing trees along a row. And that's because the fungus is moving from tree to tree through the root grafts and through the root system. So you don't need a beetle anymore. You just needed that one incursion of a beetle into your orchard to have an outbreak. So all the varieties, commercial varieties that we have tested so far are susceptible, including the king of the avocados, right? The Hass. So if you think that you're gonna grow Hass up north and that you are safe because you are not growing these huge tropical avocados, you are also wrong because Hass is susceptible. Now, there is a variation in the timing of disease progress. You can have a Simmons or a Lula that will collapse in less than a month, but perhaps you can have other varieties that will collapse maybe a month and a half, right? But that's not really, we cannot name these tolerance, right? Because it's just a matter of days or weeks. And also a lot of the rootstocks that are used commercially are susceptible to the disease as well. So we have tried many things, and many of these strategies were grower driven. So we are grateful for their input in management strategies to test. So far, we don't have any cost-effective solution, but we recommend to do extreme sanitation, removal of the infected tree from the root, and the removal of the adjacent trees, especially if that tree has advanced symptoms. So even if the adjacent trees don't look symptomatic, you need to remove them to prevent that spread along the row. We also recommend to increase the light, and so have heavily prune these trees after the production season. So why? Because these beetles like forest conditions. So they like to, to fly when there is not much light. So if you increase the light, you will decrease the population of beetles that will get into your orchard. And also tree stress. So even though the main vector of this disease is not attracted to ethanol or any of these IM6 signals, these other beetles are. So also having a healthy orchard will prevent beetle incursion into the planting. So the Tropical Research and Education Center and the University of Florida, we have collaborators in other stations and in main campus, are working night and day, or day and night, on this disease. And we have different labs collaborating and working together, including horticulture, economy, entomology, And we have a really strong extension component. We work, as you know, hand to hand with you guys, trying to find solutions for this disease. And so my lab is the pathology lab. And we are also working very hard. And so these are the main lines of research. We are looking into finding sources for tolerance, even though it's a long shot, because all the Persea genus is susceptible to the disease. You know, we may find sources of tolerance that then it can be transferred into the commercial variety. So we are still working on that. We're also trying to look for alternative synthetic fungicides. This will be a short to mid-term solution for all these people that are replanting and they will have production in 5 years, let's say. We need to have something that can protect the trees firsthand, right? Like a short-term solution. We are also looking into biological control. Of course, we are supporting long-term solutions and sustainable solutions that are environmentally friendly. So that's a line of research. And in collaboration with other labs, we are improving the detection techniques, surveillance, so we can help other production areas to prevent the establishment of the beetle and the pathogen, so to prevent the disease establishment, and also be prepared with mitigation strategies on what to do once it gets to Mexico. Because it's not if, right? When it gets to Mexico, when it gets to California and other production areas. And so, of course, I cannot do this by myself. There's many people involved in all these projects. There's many funding sources involved in this project. You guys have supported us by giving us letters of support. And we have been able to secure funding to work on all these aspects of the disease. I'm not going to explain each of them because I only been given 15 minutes, but I just want to tell you the progress that we have made. So we finished our first screening of germplasm. We made collaboration with the largest aguacate or avocado germplasm in the world that is in Mexico. As you know, that is the area of the origin of the species. And they sent us many non-commercial unique accessions from their collection. So they were kind enough to share with us the germplasm. We inoculated more than 600 seedlings, and this is arduous work. We had to wait 2 years for these plants to be able to inoculate. They had to be in quarantine and so on. So long story short, we publish our work. You can get the publication from here, or you can ask me for the publication. We found, so first of all, these are seedlings. So there's a lot of variety within families and among families. But some of the families show individuals that had very low symptoms. So that's good, right? Because there may be some genetic component to it that we can pinpoint. And so we found one cultivar that we can classify as tolerant or tolerant family, and we're going to continue to work with it. And just to give you an idea, Lula, which is very susceptible and we use as a control, is here. So that means that there is even worse germplasms. But there's also some better, right? The ones that have a lot of green are the ones that have plants within that family that can survive the inoculation. Those plants were recovered because avocado, the good thing is that it can resprout. So even though the rating is destructive, we were able to save the plants and now they are growing in the field. Hopefully we have time to see them today. And that is going to be a source for research in the future. In terms of fungicides, I'm very excited because we re-evaluated a set of fungicides. This work has been done by Klotz in the past, but a decade ago. So now we have better formulations, we have more knowledge. So I kind of started again that process. We have to start with in vitro testing, of course, at the beginning to see what works, what doesn't. And then we move into plant output. Again, long story short here, we found one compound called flutriafol that can move really good in planta. But we are talking about potted trees, right? This is the caveat. These are potted trees. But if you can see here, these graphs are residue testing of sapwood. So that I took the vascular system of the stems and did a residue testing to see how much flutriafol is present in this tissue. And you can see here flutriafol comparing to tilt or to propiconazole. So it's 20 times more mobile. And so that's very encouraging. So I was able to convince the industry, so the chemical companies, and the IR4 program to support research in the field for these compounds. I'm not saying go and buy it and go and use it, please. This has to be tested if it's not phytotoxic to the plant in the field. It has to be tested for residue in the fruit, and it has to be EPA approved. So there is a long road ahead, but we are making progress, and I'm very excited about it. So for the biological control, which I'm very fond of, we were investigating a fungus called trichoderma, which is known to be a good fungus, and it's very widely used as a soil amendment and root enhancer. And so we isolated endophytic trichoderma. So trichoderma that lives within trees. Some of the sources were survivor avocado trees. And we extracted the metabolites of these fungi, of these species of trichoderma. We tested the antagonistic effects, so how much it restricts the growth of Harringtonia lauricola, which is the pathogen. You can see here the control, and you can see here how this strain of trichoderma has 80% prevent the growth or inhibit the growth of the pathogen. So that's promising, right? But we wanted to make it endophytic again. So we inoculated the seeds of Hass seedlings, and then we waited forever, and then we tested if it becomes endophytic. And we've proven that it becomes endophytic. It was recovered from the roots, from the stem, and in one case from the leaves of these seedlings. However, in very low quantities. So it's not really a good colonizer of the seedling. Again, long story short, what we want to know is, bottom line, if we inoculate these plants with a pathogen, with the laurel pathogen, is trichoderma that is living inside the seedlings gonna prevent the disease or not? No. So all the plants got laurel wilt. They all yielded the pathogen. So it is what it is, right? However, there is some inhibition. So there are some differences. They may be doing something, but it's not the silver bullet. It's doing something, it's preventing the growth, but it's not enough. It's not good enough, right? What is next? We are gonna continue our screening. We have an ongoing second batch of germplasm from Mexico, from California as well. And we are soon gonna finish that screening. We are gonna look into the genetics of the surviving trees, and we are gonna propagate these survivors from the screening process to see how we can move forward. I'm very excited to start the field testing of flutriafol-based compounds. There is one formulation that is registered for food crops, so that's exciting because already jumped a lot of hoops, and so we'll see how it goes. And then I'm still a believer of biological control as a management tool, so we'll continue with that. Okay, so that's laurel wilt. Hopefully it was not too fast, but now I'm going to talk about phytophthora root rot. Very different disease, but very important. So it may not be as aggressive and dramatic as laurel wilt. However, it is extremely important because it's wide distribution and economical importance. So again, this pathogen has more than 1,000 hosts. So that's let it be distributed so widely, right? So virtually any production area has a problem with phytophthora, right? It can be a little problem or it can be a big problem, but it is widely distributed. Most of the work that I'm going to present has been done by my colleague Patricia Manosalva and her lab in UC Riverside.
Has any of the research used interface with artificial intelligence? Or is it all done on individual lab?
We can go back to that question. I will have to to think about it. Yeah. Yeah, I mean, we have to do the baseline first. But there has been, detection, for instance, detection projects that use AI to quantify or to detect, trees infected with that disease. But we are far away from using AI. We have to establish the baseline first. Anyways, so Phytophthora root rot is caused by, species in the Phytophthora genus. And this is not, they are not true fungi. So this is very important to know that these organisms are not true fungi. And it's important to know because a lot of the fungicides are not gonna work, depending on the pathway that they interfere with, they are not gonna work with this group of organisms. So the main difference is that they produce zoospores. So they produce spores that can swim. So when we talk about flooding, and Dr. Schaefer is going to talk about it, this becomes very important because they really like to swim and they like these water rivers that form underneath the ground. Here you can see some examples of the spores. They are able to move, they are able to swim, they have flagellum. So again, these are not true fungi, they are all mycetes. So the symptoms in the root system, you can see here, there is necrosis, discoloration, all the way to decay. And that can affect the entire feeder root system. And feeder roots are really important because these are the ones that will uptake the water and the nutrients, right? So they are really the base of tree health. And so these pathogens can affect the root system, creating lesions and ultimately killing the tissue. And you can see here some examples. The good thing is that the avocado trees have very superficial roots. So for you to inspect the roots, it's not that complicated. And you can take a look and see if they are discolored. They should be white or cream like color. So because the feeder roots are so important for nutrient and water uptake, you will see the symptoms in the canopy, right? You will see kind of nutritional deficiency symptoms. You will see yellowing. You will see defoliation, which will conduct, because if the tree is having problems with nutrient and water uptake, and on top of that is losing the leaves, then you will get the branch dieback, right? And then the slowly and painfully decline of the trees. These are kind of more like a chronic disease. And so you can see here examples of defoliation and branch dieback. Now, in young trees, for people that want to start a new planting, it's important to take a look at the presence of Phytophthora, right? Because it can, when you put a young tree in the field, it's going to struggle anyways because you are transplanting the tree, because it's a new environment, because it's a young tree, not very, very, healthy. And so if you have Phytophthora on top of that, you may see outbreaks of this disease and ultimately the death of the transplanted. So Phytophthora is the excellent example to talk about the disease triangle. And this is what plant pathologists live by, right? Basically, it illustrates the three components that have to happen for disease to occur. You have to have a susceptible host, you have to have the pathogen present, and you have to have a conducive environment for the pathogen to infect, colonize, and reproduce, okay? So avocados are susceptible to Phytophthora. Phytophthora is ubiquitous, really, in the soil. And we're going to talk a little bit, and Bruce is going to really dig into the conducive environment for this disease to establish and to spread. And so again, I'm not going to talk about that, but I just wanted to say that because of the importance of this disease and how long we have been dealing with this disease, there are a lot of strategies out there. You can look in avocados from Australia. They have this really nice poster with showing what is called as a peg wheel. And then this kind of illustration gives you all the strategies that you can use to have an integrated pest management approach. So for Phytophthora, we have a lot of options, and we should be using multiple options, right? And so they are basically affecting or targeting one of those components of disease, either by cultural practices or by using tolerant material or by using chemicals to affect the pathogen. So why do we care now? Phytophthora have been around for a long time, but now we should care more. One, because flooding events, and again, Bruce is gonna touch into the flooding factor, how it contributes to the spread of this disease, but the water table is coming up and we have more torrential storms happening. So there's a lot of more flooding. So we now should care more about Phytophthora. We need to keep the trees healthy because we don't want ambrosia beetles to get into your orchard. So by having healthy trees, we will avoid that. And of course, we need to have very productive remaining acreage of avocado, right? Okay, yes, we have lost a lot of acreage, we keep losing trees, but the ones that we still have, they need to be top shape, right? And very productive. And when you have Phytophthora infection, the yield goes down. So Jonathan and I and people from my lab did a survey just to know what's going on, not only in southern Florida, but also in central Florida, since you guys are so interested in expanding the avocado industry. So we went to several places, we dig roots, we collect the roots and soil, and we ended up with 75 samples. We sent them to our colleagues' lab in UC Riverside for analysis. And we did find the presence of Phytophthora in central Florida that was confirmed by our colleagues. But also we saw a lot of Phytophthora-like symptoms, but no Phytophthora. So that is, I mean, that's something that is another thing that we need to research, because I think there is something else going on that we are not capturing. Okay, so to summarize, what they found is that the Florida strains of Phytophthora grow better in warmer temperatures, you know. So, I mean, that's kind of expected, but not all of them. So there was a variation in how good they grow depending on the temperature. They also found that the Florida strains, in comparison to the other strains collected in other states, some strains are less sensitive to phosphonates, and that's, don't panic, still works, but we have to look into the future, right? Because that means that we are selecting for strains that are not that sensitive anymore to some of the management strategies that we are using. So we need to layer strategies to avoid resistance. So that's the main message. And my colleagues or lab, Patty's lab, is still working in a lot of different venues. They are testing the sensitivity towards Ridomil and some of the other fungicides that we are using. So we hope to have that data for you next. Okay, so that was the two major diseases that I was given 15 minutes to talk about, probably more than 15 minutes. But anyway, so thank you everybody that gave us letters. Thank you everybody that let us go inside your fields to test, provide samples for us, and all the support. Time for questions. Yeah. Okay.
Good morning, everybody. I'm the plant physiologist here. I have 100% research appointments, so I don't get out that often in front of growers. Jonathan and Ramina make me every once in a while. Anyway, I'm going to talk about flooding and phytophthora root rot on avocado trees. Flooding and waterlogging of agricultural fields are increasing in Florida, as you can see from this aerial view of the agricultural area of Florida. After just a few days of heavy rains, you can get quite a bit of standing water. Flooding refers to water that's above the soil surface. Waterlogging, which is just as much of a problem, means that all the soil pore space is full of water. And when I talk about flooding and waterlogging, the damage from this to the plants is due to lack of oxygen. Too much water is not a problem to the plants. The problem is that when soils are flooded or waterlogged, oxygen diffuses 10,000 times slower in water than it does in air. So the roots are not getting enough oxygen for normal function. So the agricultural areas of Miami-Dade County, where most of Florida avocados are grown, are very prone to flooding. The reason for this is everybody knows that grows down here. The soils are very porous. The water table is very close to the surface, as you can see from this canal. This was taken during the dry season. During the wet season for this watermark, you can see the soil comes even closer to the soil surface. So just after a few days of heavy rain or tropical storm or hurricane, the water can easily rise above the soil surface. There's the Everglades Restoration Project, which many of you are familiar with. We've been raising the water table more and more for several years, and the adjacent agricultural area is becoming more and more prone to flooding because of that. And then finally, as a result of climate change, the occurrence of tropical storms and heavy rains and hurricanes is increasing throughout the world, and that's true in Florida, not only in South Florida, but all parts of Florida. And this is becoming a problem in Central Florida, where some avocado production is taking place. And so some of you have seen this in your own groves, that just a few days of heavy rain and avocado growth will cause standing water. Now, the soil type affects avocado tree responses to phytophthora root rot or flooding. And the picture at the top is a few different avocado groves in Chile. And you can see spots where a lot of the trees died after a while. And they thought this was phytophthora at first. These soils are really poorly drained, especially in those areas. They sampled these groves for phytophthora and they didn't find any, so they attributed that to a really poor soil aeration. On the other hand, when there's decent drainage in the soil, but the soil has a good water holding capacity, such as in the bottom, that picture was phytophthora root rot damage in a soil that's normally has pretty good drainage, and there's no waterlogging or flooding. Now, in the chrome gravelly loam soils of Miami-Dade County, these soils, unless we have a flooding, soils are generally well drained. Phytophthora, there's been sampling, and I don't know what these guys sampling found. I don't know how much phytophthora found, but many years ago, Randy Pless did some sampling, and he did find phytophthora in these groves. I assume you guys found it as well. Now, this was an avocado grove in Miami-Dade County, and this was taken in 1981 after Tropical Storm Dennis. And many of the trees in these groves had died after there was standing water in the grove, depending on the grove, from 3 to 5 days. So everybody thought, well, maybe this is phytophthora. And at that time, there had been lots of research on phytophthora effects on avocados, but there was really no research on flooding. A couple of years later, Randy Pless, who's now retired, a plant pathologist, came to Trek, and I came to Trek here, and we decided to do some research on the interaction between phytophthora and flooding on the effect of avocados. Now, first, I want to just mention that flooded trees often don't show any visible signs of flooding damage until weeks after they're flooded. So often they'll look great, the soil will be drained, and then all of a sudden, several weeks later, if they're stressed, often they will die. So we needed a way to measure stress that where you can't see any physical signs of damage. So by using this portable gas analyzer, we can measure photosynthesis, also called net CO2 assimilation, which we found was a very good indication of plant stress due to several factors, and flooding and phytophthora is one of them. And usually if the photosynthesis is very low for a period of time, the plants will eventually die. We did a lot of experiments with different age trees and different environments with potted plants. And this over here, this is one of our first experiments we did, and this goes back 35 years ago, where we had photosynthesis on the y-axis, and this is, we infested the soil with the phytophthora. So on the top was a control where there's no phytophthora infesting the soil, and this was not brome soil that we have in Florida, but this was a soil with a very good water holding capacity. So we can see as the leaves aged a little bit, we got a little bit of increase in the control and photosynthesis. But when we infested the soil with phytophthora, very quickly we got a drop in photosynthesis. Then after a while, we flooded plants in each group, and we had so much damage from phytophthora in the infested treatment that we couldn't see any additional effect in this soil of flooding. And for the control in this particular soil under this environment, 7 days of flooding didn't really do too much to the trees. Well, then we did some more studies looking at what happens in chrome, very gravelly loam soil. And so what we did again, we infested the soil with a pretty high level of phytophthora. And over here, the dotted lines is the infested soil, and the solid line is the non-infested soil, the control. And we can see even after 42 days, these are not flooded. We can see that there was really not much of a difference in photosynthesis, and these plants all look great. And then we flooded some of the plants for a few weeks. And so if we look at the top two lines, those were the plants that were not flooded with and without phytophthora. We can see after 70 days in chrome soil with these small potted plants, we don't see really any much of a difference between the phytophthora infested and non-infested, which was very different from what we saw in the soil with a very normal, very high water holding capacity. Now over here, the line with the triangles, those were flooded plants that did not have phytophthora. So flooding by itself with no phytophthora is damaging to avocados and chrome soil, but it takes a while. And again, that depends on the age of the plants and the temperature, but it still takes a while compared to plants that had phytophthora that were flooded. And the photosynthesis shut down the first time we measured it, and those plants all died. These plants eventually died too. So the bottom line with this is in chrome soil, phytophthora root rot, although it exists there, it's usually not a problem, but flooding alone can be. And there's a strong additive effect between phytophthora and flooding in these soils. And we also looked at this another way. Okay, what we did is we infested the soil with different levels of phytophthora, and we measured different amounts of root damage. And then we either didn't flood them or flooded them. And we looked at the amount of root damage by phytophthora, how it relates, how the amount of root damage affects the response to flooding. So the line at the top is plants that were not flooded, and this is on the bottom is the amount of root necrosis from phytophthora. And this again is net CO2 assimilation or photosynthesis as an indication of plant stress. And we could see the photosynthesis was reduced when we got up to like 80% root damage from phytophthora. But even with 80% root damage in this soil, which is very surprising, the plants are still surviving, they're still photosynthesizing, although at a reduced rate. However, just about 30% of root damage from phytophthora with flooding, and the photosynthesis stopped immediately. And plants with most of this amount of root necrosis from about 30 all the way to about 90 when they were flooded. Not only did photosynthesis stop, but they also died eventually. So again, there's a very strong interaction in chrome soils. Phytophthora is not as problem, but there's a strong interaction between phytophthora and flooding. And that's going to be more of a problem as flooding becomes more of a problem. So now, I don't have 15 minutes. I'll talk briefly about some things that can be done to mitigate flooding and or phytophthora damage. For phytophthora alone, there's several things that can be done. Phosphorus acid as an injectable fungicide is common in many places in the world, and we're going to talk briefly about that. But I'm going to talk mostly about flooding and phytophthora. So a simple solution, of course, is in soils where flooding is a problem and maybe phytophthora, is not a problem, because you can just plant trees on hills. Like this picture was taken in Chile on these little mounds, or this is taken in Dade County, or you can plant them on, on beds. Problem with that though, is even when you have flooding, the soil can still come up through the beds and capillarities, so that might not be the best answer. Now there are phytophthora tolerant rootstocks that are available. These are commercially available. And the most common ones are Dusa, Toro Canyon, and Duke 7. And in areas where phytophthora is a pretty big problem, such as California, it's common. They have different amounts of phytophthora tolerance. Dusa is a little better than Toro Canyon or Duke 7. But there's problems with all of them. All of them are susceptible to waterlogging, for example. And so recently there was new phytophthora tolerant rootstocks developed by Patty Manasalvo's laboratory at the University of California, Riverside. These are not commercially available yet. Our plan is to make them commercially available eventually. But these are the ones that, these are the names of them. And we have currently have a project, a collaborative project with Patty. Several of us here are working with her. And we're testing these rootstocks on Haas, Jam, Reed, and Donnie Scions. We haven't planted in the field at Trek to look at the horticultural characteristics, and Edwin is going to talk more about that. And we also were testing them. We had tested them in the greenhouse, and we have one more batch to test for susceptibility to laurel wilt. Because even if we find that they do well here, they're just very susceptible to laurel wilt. It's not going to be very helpful. Okay. Also, many years ago, we looked at fungicides to improve tree recovery from flooding. In soils that had phytophthora. And again, we have photosynthesis on the y-axis. And what we did is we either injected the trees with injectable alliette. This was done about 35 years ago, so it was before phosphorus acid was we could use phosphorus acid to inject into the trees in the United States. But injectable alliette is very similar to phosphorus acid. It breaks down phosphorus acid in the tree. So we looked at phosphorus acid and Ridamil two ways. One, prior to infesting the soil with phytophthora as a preventative, and another way after we infested the soil and planted the trees as sort of a control. And then we flooded the trees for a period of time, and then for 7 days. And then this graph is after the trees were unflooded. And what we looked at is recovery from flooding and from photosynthesis. And this was very hot greenhouse. So in this greenhouse, and this was in chrome soil. So in this greenhouse, this is the control. There was absolutely no recovery when we just did not put any fungicide. When we used either fungicide as a control after the trees, were planted in infested soil, there was no recovery. But with Ridamil as a soil drench, the plants did recover from flooding. And when we injected alliette prior to infesting the soil, we had a pretty good recovery of the plants after flooding. So the flooding stress hurt the plants, but the trees were able to recover from 7 days of flooding. Whether this is practical, probably not in South Florida, because basically phytophthora is only a problem when there's flooding. And you don't want to spend the expense and the time of inoculating or injecting your trees with phosphorus acid. Now it would be phosphorus acid rather than alliette in the case that you might get a tropical storm or a hurricane. But it was just interesting to see this. Another thing that can be done is fruit removal and pruning within a few days after flooding. It reduces the transpirational demand on the stressed roots and increases tree survival. Especially if there's phytophthora, the roots are already damaged somewhat from phytophthora, the flooding damages them more. And so there's usually not enough, usually the canopy puts too much stress on the damaged roots. But if you severely either remove fruit if there's fruit and severely prune the trees, that has been very successful. And what we recommend is to remove all fruit if they occur and to severely prune a tree from like 1/3 to 2/3 of the canopy. What we thought also is after a hurricane or tropical storm, everybody's very busy. So it's often not practical to go out and prune trees because people are busy working on their houses. And so we thought, what happens if we know a hurricane or tropical storm is coming? Maybe if people have time, they can go out and prune their tree ahead of time. That didn't work out so well. We found out that pruning and fruit removal must be done immediately after flooding. If we prune shortly before flooding, it actually increases flooding stress and mortality. And we did a lot of research and we know the reasons for that, but I won't go into that since I only have 15 minutes, but I'd be glad to discuss it with anybody, the reasons for that. Okay, now there's some potential approaches that we're looking at that aren't, that are definitely not in use now, but they might have some potential. One is we tested just a few years ago, we tested the effects of steroids on recovery from flooding. Steroids are a class of plant hormones that have stimulated plant immune responses to several different stresses. We tested 2 different steroids applied as a soil drench prior to flooding avocados. These were the 2 tested, 28 homobrassinolide and 24 epibrassinolide. And we took a lot of different physiological measurements, but I'll just show you the tree survival. Here is the non-flooded, and the most important thing is the flooded trees. This is after flooding, after we drain the trees from flooding. We can see the control. This was several days of flooding or a couple weeks of flooding. After the control, only 20% of the trees survived. The trees treated with homobrassinolide, 40% survived, and the trees treated as a soil drench with 24 epibrassinolide, 50% survived. So this might have some potential to be looked into a little bit further. There's also some potential for breeding for flood tolerant avocados. There's not been much work at all on breeding flood tolerance. A couple of years ago, we did some work looking at clonal avocados and looking at the different races. Most of you avocado growers know that there's 3 races of avocado: the Mexican, Guatemalan, and West Indian, depending where they're from. The West Indian, that's a misnomer. They're really from the lowlands of Central America. But what we found is there's a difference in flooding tolerance among the avocado races, at least the cultivars we tested, the clonal cultivars we tested. With the Mexican cultivars appear to be the most tolerant of flooding, and the Guatemalan cultivars appear to be the most sensitive. And again, we didn't test all cultivars. We only tested a few representative ones. That's what we found with what we tested. And one of the reasons for that, at least, plants often respond or mitigate stress responses by producing antioxidants. And we looked at we separated, this is called a principal component analysis, but we were able to separate the different cultivars based on the amount of antioxidants they produced in response to flooding. Prior to flooding, we couldn't separate any of the cultivars. But after flooding, we can see that the Mexicans produced much higher levels of many different antioxidants, whereas the Guatemalans produced much fewer in response to flooding. So that's one of the reasons for attributing the difference in flood tolerance. And so this has implications for potential breeding avocado for flood tolerance, although that's a very long-term solution, unfortunately. And then the last thing I want to talk about is biotechnology approaches, which there hasn't been much done lately with this. This was work done many years ago by Richard Litz, who was a tissue culturist here. He's since retired. But avocado compared to other fruit trees is very flood sensitive. Probably one of the most flood sensitive of fruit trees. There's a few forest Persea species, which is in the same genus as avocado, such as Persea borgonia, Persea cinerascens, and Persea palustris. Persea palustris is even called swamp babe because it's a common name because it can grow in swampy situations. These plants are moderately tolerant of waterlogging or low soil oxygen, and they're also somewhat resistant to phytophthora root rot. But the problem is they're not sexually compatible or graft compatible with Persea americana, with avocado. So what Richard did was he isolated protoplasts, single protoplast, and through a process called protoplast fusion, he fused the protoplast, then grew the plants in tissue culture, and he was able to combine these phytophthora-resistant and somewhat flood-tolerant avocados with sparge species with avocado, and then grow the plants up in tissue culture. At that time, then our next plan was to screen them for flood tolerance and look at phytophthora tolerance. The problem is at that time, this was about 20 years ago. At that time, we did not get good survival. We got a few plants to survive, but not at all enough to screen them even in pots. And so that's, Richard retired and this work was stopped. But now, there's been a lot of work with tissue culture of avocado over the years, not for flooding roots, but just to produce them. And there's actually a few labs around the world that produce avocado through tissue culture commercially. So this would be really interesting, I think, to start picking this up and actually for some of these labs to start looking at it in the future. Work. And that's basically all I got. Thanks for your attention. Any questions?
One sentence punchline. Why is it that trimming them before flooding makes it worse?
Because we're reducing the photosynthetic area by taking the leaves off.
Okay.
And what happens is when you reduce the amount of carbohydrates that go to the roots, and the roots go into anaerobic respiration under the flooding, and they need a carbohydrate
no
substrate, so you're reducing the substrate for anaerobic respiration. And we got lots of data on that, but I don't want to bore this track.
Yeah, it's just not intuitive. Thank you.
Yeah, just not. That's why we were surprised.
Hey Bruce, 2 questions from online. One is from Nitsa. Can these steroids be purchased?
What we used was, we're chemical, we bought from a chemical supply company. So as far as I know, there might be some product, I think there are some products that are available. I don't know what they're registered for. But as far as the 2 we use, these were not, commercial. These were just, it was just, this was a test of concept,
Right.
more than this.
Got it. And then Wan Chan says, what's the application concentration for the steroids?
I can get back to him on that. If he sends me an
Okay.
email, you can send him my email because I don't have it right in my
Yeah.
head, but we do.
Sure.
We have published that in a journal called Horticulture, and that's in there. So if he
Okay.
looks at, if he types avocado brassica steroids, journal Horticulture, it's in there. And then he doesn't.
Thank you, Bruce.
Any other questions? Okay, thank you.
You take my time's very well. No. So, good morning. Thanks very much for being here and people joining us online. My name is, I am in the age that I don't know if that works or not. My name is Edwin. I'm working as a postdoctoral researcher since 2022. And after that detailed explanation about what those diseases are, so I'm gonna focus on what we are doing in the field and greenhouse as well, testing those materials that Bruce mentioned. We basically are comparing some science and rootstock interaction. So you're gonna see mostly here what we have been doing in the field, and briefly mention about we found in the greenhouse testing laurel wilt tolerance, which is also important because, as you can see, we can separate. So we have farmers, we have growers, and we have many products, and we need to put in the same pocket. So that's the thing, and that's we are showing here. So my presentation gonna be splitting 4 chapters: introduction, how we establish the field that you're gonna see later after this presentation. Some preliminary results. Those are 2.5 years results, so it doesn't mean that we have conclusions, but we have something that we may we are seeing something, and final comments. Again, this is a project funded by UC Riverside, USDA. This is the grant number and the information you can see here, and the UF at track. So this is basically what we did. Or we did since we started in May 2022. We got the plant from California. It took like months to get the plants here, to receive, to prepare. They came in a cooling transport, so we need to wait, because they need to adapt in the Florida conditions. So after we got the plant ready to start, we started like splitting in groups and also in the field. So that's why you're gonna see some slight differences in the planting in the field. And this is what we have. We have 4 Scions. Reed, Haas, Gem, and Donny. Those are, I'm not sure if Gem is commercially available here, but so those are like the commercial side that we are testing. Combined with those 6 rootstocks, I don't know, for some reason it's showing the format out, but you can see this PP35, PP40, PP42, 45, and 80. Those are not commercially available. People from California want to remark that those are being tested, and the only commercial one that we have is the Walding. So from planting until now, this you're gonna see in the field is the map. Those plants are distributing blocks. So we have, you can see here from 1 to 8. You're probably gonna see the 8, but there's some problem with the map. So we have 8 blocks, and you can see those blocks or those plants on green. They were planted little bit before. So we have basically 3 months difference between the planting between the white and the green. That's not too much, but and this is the map. You can see in the field, you're gonna see these marks, meaning what we have as a Scion and rootstock. So for example, this Gem plant, Gem Scion on PP80. This is the plant on the row number 1, and in the lines number 12. So just for information purposes, you're gonna see the field much more better. This was in 2022 when we planted the trees. You can see the soil, how it basically is, and how we prepare the holes and the field. You can see here once we finish. And then after that, we start putting the irrigation system, putting the plants, planting, tying into the stakes, because basically those plants can be stable by themselves. So we need, especially with the clonal one, we had issues at that time and for around 6 months to get by themselves stable. So we also stake most of those plants. With the Walding, we didn't have that much problem, but with the clonals, we used to keep them, pulling up and helping to keep it straight up. We started with barrier irrigation, and we planned how can we manage the plants. Basically, it's a general program with fertilization, some solid and liquid as well, especially for iron. Mowing, how often we did, irrigation system. This one specifically, we use this smart app. It was developed by, I forgot her name, but this is available. It's an app that you can use to track and to adjust the irrigation system. Because once they start grow, we start adapting and replacing and adjusting the sprinkler to give better amount of water and better distribution. And this is basically the plan that you can find as well on the handout that you're probably gonna have on the website. How we fertilize, how we spread the fertilizers. We use for solid this machine. We also did by hand and the iron mostly by tractor with the water. So far, most of the problems with pests and disease that we got was with mites and those laced bugs that you can see on your right. Mites, especially at the beginning. Now it's a little bit less, but we still have some plants with those problems. This specific symptom we saw mostly in Haas. We don't know exactly what's going on. We may have some ideas, but you're gonna see better in the field with especially with the Haas plants. So curving, and if you see those things are not like homogeneously distributed in the canopy, but some new leaves and some branches you're gonna see better in the field. Wind damage, especially hurricanes and those kind of things that are pretty common here. So especially at the beginning, we need to take care with the plants to be really support to avoid any problem at the beginning. Once they get established, we don't have that much problem, but especially at the beginning with the clonal plants, we had those issues. So with that, I'm gonna show you briefly some results. And for that, you can see that we use those two scales for health from 1 to 5, meaning the 1 is the pure or the diet plan, and the 5 is better. And for the what we call phenology, we basically had 3 main or 3 points. If the plant is exploring, or the plant is like quiet, or if the branches are either having new leaves or having flowers. So this is the scale, meaning that the 1 is the plant is quiet, and 5 is flush, either flushing or flowering, or both as well, as you can see in the middle. So for health plant, or how they look, the plants, once we started the plant, they were pretty well. The first month after we got the first wind problems, and then in 2024 in February, we saw some differences in health that you still gonna see in the field. Some branch dying back, and some issues with those plants growing slower than the others. And for now, in 2024, now you're gonna see in the field, and you're gonna have a better idea how they are likely recovering, especially the plants, the has science. Blooming, this is interesting to see here, for example, we saw differences in science, in rootstocks, and in the interaction between some science going better with some specific rootstocks. So for example, here you can see gem and has, they start blooming this February in 2024, this year. Most of those plants, 5 means all of the plants are flowering, but 1 month after, we basically got some differences, especially in this case for gem. Those plants are on Walding showed like more blooming, and the other basically lost many of the capacity or the sperm that you can see, or either compared to flowers. So you can see, for example, gem here as well on has, it was like more or longer the period of blooming. On reed, they start late, and after 1 month after, we saw most of the plants blooming, but you see like again a short video with that phase. This is for Donny. This was longer, but 2 months after, we saw like they changed for the next phase. Once the plant, you know, in avocado we have like a flush, then a flower, then comes another set or another flush of leaves. So that's different between the varieties. For flushing here, again, you can see differences between gem and has. Some seem to be longer, or they're short, even other shorter than the others. But for example, this is for reed and for Donny. Again, reed, as you saw on the blooming, like shorter or starting later. It probably means that we're gonna have fruits later, or a longer period of harvesting. This is just a brief mention about what, because everything is important, but if we don't have fruits, it doesn't matter too much. So this is a comparison between July and September. You can see here for every group means a Scion. So you can see here, Danny, for example, has gem and reed on each rootstock, which is interesting so far to see. This is our first like official production season. So you know that it may change once the plants get older and better established. So what we saw so far, Donny, we got a lot of flowers and fruit set, the smaller one, but those 2 months after, we basically we didn't support the fruits, so except for PP on 80. The same, if you see there is a slight pattern, like same Donny has and gem and reed, you can see the PP 80, especially in those three, like little bit like same have or supporting the fruits. With the star, you can see those differences, especially for Has and Donny. But if you compare what we got in July and what we got in September, Hass, gem, and reed seems to be more stable. Again, this is, if you remember, we are using the same management, the same amount of water, the same amount of, the same management. So we may have some artificial, some, differences also due to the management, because you're gonna see down here, reed, they are taller, bigger, stronger, so we may adjust this requirement to better support of the fruits. Some preliminary results to writing up my presentation. You can see here, this is after 10 days. Basically, we see what we call the scale rate, or how fast the disease progress along the time. And with the plants, with the comparison that we got so far, with those reed and Walding, Hass on PP45, gem on PP45, gem on PP80, and gem on Walding, we saw a little or slower disease progression, which means like how fast the plants are showing the symptoms. This is a slower comparing with this like more rapid disease. So you can see like the velocity is mean like how fast the line comes up. So far, this is our first comparison, and those are the 5 combinations that we saw maybe slower compared with the others. So with that, this is mostly what I had to present. Those are people that are helping us. As Romina mentioned, this is a lot of people helping and supporting the amount of things that we need to do in the field. Preliminary conclusions. So again, we need longer and more years to have better understanding of what's going on in the field. We can see some differences among the combinations. We don't have the data to support which one specifically, but we may see that, especially since they are new rootstocks, we may have some rootstocks that may support better the production. And of course, if we find some tolerance to laurel wilt and to phytophthora, it will be nice, or it will be better to have longer information. Maybe some limitations, as I mentioned, based on the management, we're going to adjust that based on the canopy and the grow of the plants. As I mentioned, if we are intending, this is new, and John is going to mention that, if we going to establish a clonal plot or clonal orchard, we're going to need to take care with those plants, especially when they come or they arrive, because they seem to be weaker at the beginning. So these are extra lot of amount of job to do in the field. So this is important to consider because the cost and the process to establish plants. A final thing that you're gonna see also in the field is that the plants on Walding seem to be lower branching. They have like more, and the branch comes from the ground more. This could be good if we think about the distribution of the canopy in terms of management. But again, we need to see that at least 10 to 15 years. So that's my presentation. This is why it's important to keep researching in avocado, and thank you so much.
Anyway, when you talk about Waldings, you're talking about Walding seedlings.
Seedling, yeah,
Not
thank you.
clones.
No, thank you. No, those are also seedlings.
Do you have any self-grafted plots in there, like control?
Where in the field?
Yeah.
You mean like constant?
Like self-grafting, not on Scion stuff.
No, our plot. Jonathan you're gonna see we have other plants that are not part of the project, but those plants are clonal, except the Walding, which is seedling, but they're basically the same. Any question online? No? Okay. Any other question?
Yeah, just follow up. From what I saw that your research indicates, seems like the Walding seedling just do as well as some of the other clonals.
Yes, so far yeah.
Remember, those trees are only about 2.5, 3 years old.
So yeah,
To
so
determine
far,
which is gonna end up best. Within the 2 years,
Mhm.
they're looking back.
yeah. And you're gonna see by yourself in the field, so have you better conclusions?
He's gonna take you out to the field if you want to see results.
So what's the recommended rootstock then?
Well, no,
It's more complicated than that. Remember, he said those rootstocks are not available.
Yeah.
There are clonal rootstocks from sort of the first generation of phytophthora root rot resistant rootstocks, Duke 7, Toro Canyon, a number of those which were developed in California. They are available, but the catch is there's no nursery here producing those clonal rootstocks. So if you want trees on those rootstocks, you need to order them from California and ship them from California to Florida. Very expensive. The trees are expensive and then the shipping is just outrageous. So I'm gonna talk about that.
Yeah,
So,
you can start your
let me just
presentation.
go through this. Yeah, I've already finished. So anyway, I just want to talk a little bit about potential risks and opportunities and issues with planting avocados in and outside of Miami-Dade County. I want to acknowledge all these researchers from Florida, California, Hawaii, Texas. We've had a large group of people working on phytophthora root rot and also on laurel wilt, and so I just want to acknowledge that. And we've had an advisory panel with this previous grant, which is just ending. Okay, so this is just me and ranking the risks currently as I see them. So in Miami-Dade County, laurel wilt, huge risk. Hurricanes, flooding, phytophthora root rot, and freeze events. And I put freeze events at the end. Why? Because we are now a tropical country here. Because if you look at the weather data, the climate data, it's definitely showing this trend in the past 10 years. So doesn't mean we can't have a devastating freeze, but probability is going way down. Outside Miami-Dade County though, even though a lot of people think that global warming freeze events are over with, much greater risk outside of Miami-Dade County to our north. Martin County, Okeechobee County, Polk County, all those counties. So I would say for avocado production, well, for tropical fruits really, but for avocado production, freeze events are still going to be a major thing to think about if you're going to plant outside Miami-Dade County. Flooding is another. So there are people putting in groves in what we would call historically the flatwoods area. These were the flatwoods area where citrus groves were planted in low-lying land that would flood. And so people put in the infrastructure to plant citrus trees on beds. So they had extensive bedding systems with extensive drainage systems. So the land was contoured, the beds were contoured. In some cases, tile drains were put in. Most cases, they had pumps to move water off the property if they were having a high water table. And so we have new people getting into avocados and other fruit crops up north, and they're using that land, but the infrastructure may not have been maintained recently, or was not implemented or maintained. So they come in and they plant avocados, and they have flooding events, but they don't have the pumping capacity to move the water off, or the contours on the land or the ditches to move the water off. So that to me is like the second biggest thing. If you're going to try to grow avocados on these sandy soils, a simple thing too is a lot of those sandy soils, not a lot, a number of those sandy soils have what's called a hard pan. So they'll have anywhere from a foot to 3 feet or 4 feet of sand, which drains well. But below that, inside the profile is a hard pan of different density soil, and it impedes the drainage of the water. And so what you don't see on the surface, you don't see it's flooded on the surface. Actually, down below where the root system is, it is saturated at low oxygen conditions. And so then they run into problems. So I would say along with the flooding, phytophthora root rot would be my number 3. I may have to change where number 4 is because hurricanes apparently have decided to crisscross the entire state now. But I would say hurricanes would be number 4, really. You can prepare for them depending on the hurricane, depending on how significant a storm it is, or how many you have in 1 year. And then laurel wilt, and Romina mentioned this, others have mentioned this. So some of the plantings I've seen up north, they are surrounded by native forests. And as we mentioned before, there are native trees in the laurel family, the Lauraceae family, avocado relatives, that are highly susceptible to laurel wilt. And so those trees are at risk, but that's also a reservoir for a potential grove that's put up in that area. So admittedly, most of the groves that are up there, I think the oldest one I've seen, well, and there's some, actually I've seen one that was probably 30 years old, but small planting. Some of them are, a lot of the ones that I'm most familiar with now is probably about 7, 8 years old at most. So the trees are still relatively young. So probably not as attractive to ambrosia beetles as, you know, the bigger older trees. However, if they are flood stressed, they will produce ethanol, and ethanol is a universal attractant for ambrosia beetles. So you're increasing your risk of attracting these beetles even to your younger trees, which normally wouldn't be infested, right? So it's just something that people need to keep in mind. You know, we do have some strategies, some passive strategy that helps tamp down beetle activity in an area, and that has to do with pruning. As you guys know, these beetles are forest, they evolved in forests, they like shade, and they don't like wind. And so that whole idea with us recommending that people top their trees and prune their trees, top and hedge, or hand prune, whatever you want to do, is to open up the canopy to additional sunlight, more hours of sunlight and more intense sunlight, but also it opens up the canopy to more wind movement. And so remember, these beetles are like 2-3 millimeters large. And so it doesn't take much wind to like, so they don't like wind. They mostly fly at dusk, somewhere between 4 and 8 p.m. Let's see. So ranking of opportunities to go in a different direction. So in Miami-Dade County, hopefully, you know, reestablishment of production with this replanting that's been happening in the past 5-6 years. We do have an advantage in that we already have established packing houses here. And they have established markets. I know our production is way down, but hopefully with these new frees, we're gonna start to see that tick up some. I do realize that the competition from offshore is intense and is eating into some of our season, especially in the fall. Right now, not as much early, but in the fall. We do have an advantage in Miami-Dade County is that the growers here, they have the cultural practices know-how. They know how to grow avocados. And we generally have well-drained soils. Outside of Miami-Dade County, some of these groves, they are establishing, or they have established these groves with alternative markets in mind. So some of these people are growing Hass or Hass-like varieties because they want to go for a different market. They want to go for the Hass market, but the idea is that it's going to be an alternative to bringing in fruit, especially from offshore. The idea. And then, as I said, there's interest in some companies, actually, there's a number of companies that are interested in increasing Hass production in Florida, mostly to probably service the eastern seaboard of the US. I mean, there's absolutely millions of tons of avocados coming in from Mexico, but they're having a lot of social issues, political issues. I mean, you guys have probably heard in some cases where the inspectors for avocado shipments to the US are threatened with that, you know, these avocados are okay, they're good to go. So this USD has pulled the inspectors out and said, well, we're just not going to inspect them. That means they can't ship them to the US. And that's happened a couple of times. So it's a mixed thing. And so some of the companies have said, hey, we need a reliable source of avocados. And then also, as I mentioned, these younger trees tend to be less susceptible or less attractive to the ambrosia beetles. Again, though, they've got to be healthy. So anyway, I won't go through all of this in total detail, but again, the risks in Miami-Dade, as I mentioned already, and outside Miami-Dade County. And again, the freeze events, I think, is really the big one. And part of the problem with the freeze events is number one, you need to try to find as warm a site as possible. But here in Miami-Dade County, we can use high volume irrigation to protect the trees from a freeze event. And I'm talking about
quarter-inch of water per acre per hour. So we have access, we can get those permits to use that much water for a freeze event. But in most, and I'd say most of the counties, many of the counties, especially on the west coast, parts of central Florida, even on the east coast, you can't get a permit for the amount of water that you need to protect the trees like we do. So you can get permits to use for microjets and things like that. Well, microjets, don't put out nearly as much water in general as what we can with our overhead or high volume under tree sprinklers. So people need to really think about, how am I going to protect these young trees? Because admittedly, as the trees get larger, they will be more tolerant of a freeze, but you have that 8-10 years to get them larger so that they're more tolerant. See what I mean? In summary, so, there is this potential for high water tables and flooding events. One thing that I think is, if we really want to see people using phytophthora root rot resistant rootstocks, we really need to encourage some nurseries in Florida to begin that process of learning how to make local avocado rootstocks. And begin propagating them, because getting them to California and shipping them here is just outrageously expensive. And again, I would say anybody who's gonna plant avocado trees, whether it's here or outside of Miami-Dade, they really need to implement the soil drainage strategies, including bedding, and they need to prepare for pieces. And I also, before I forget, so Romina mentioned briefly, so we've had a number of grants, and we just recently got another grant on laurel wilt. So we're going to be going in some new directions and perfecting other directions for trying to figure out how to mitigate or control it. Let me see, and I think that's mainly it. So if there's any questions, and if there's no questions, yeah. Yeah,
We heard you talking about Haas, as if it would be a viable option in Florida. So my question to you is, do you know anyone that is growing Haas successfully to be sold commercially in Florida?
Yeah, there's some people in Martin County, but it's a small amount they're growing. The history of Haas in Florida.
It's gone. It's
You're talking about the Del Monte plant?
it's
It's
gone.
Yeah, but I'm talking about somebody else.
Oh, okay.
There's another guy that's growing. Yeah, the Del Monte plant.
You can grow them, but the question is whether successfully
Yeah,
have been
yeah,
sold
exactly.
commercially.
Yeah, so the history of Haas in Florida is that it does not grow well here, that the fruit tends to be small, doesn't mature well, and all this, right? So yeah, I know somebody up north that is growing it and selling Haas and lamb Haas. And a few other things that they're growing. It's a small planting. I think he's got 20 acres. But having said that, we have been evaluating Haas here, and I'm not recommending Haas for Florida. We've been evaluating Haas down here. And like I said, the history of Haas down here is that it just doesn't do well. The tree is not adapted to this type of climate. But we've looked at gem Haas, lamb Haas, harvest, and Gwen. And right now, at least currently, it looks like lamb Haas out of those Haas-like varieties seems to be the best one. But we're gonna get ready to publish that, but not Haas. Yeah, Carlos, I mean, right.
Is that why you had it in quotation marks on your?
No, quotation mark? No, it's a single quotation
Oh,
mark.
single.
When you write a cultivar name, you're supposed to put it
Oh.
in single, yeah, you're supposed to put it in single.
Paying attention.
That's, yeah, yes, Mark.
I know you've mentioned salinity issues here and relating to the water table. But you didn't really talk about it now.
Yeah, so salinity, could be an issue, we know, the saline line in Miami-Dade County has moved in a mile since 2011. And, we can only raise the water table so high to keep the salinity out before we're standing ankle deep in water. Oh yeah, it's not saline, but we're standing in water. And then other parts of the state too, along the coasts and other areas could have salinity problems. Turns out that, West Indian, if you look at the literature, that, avocado rootstocks with West Indian background tend to have more tolerance of salinity than others. And there are clonal rootstocks with salinity tolerance. But again, nobody is producing clonal rootstocks in Florida, so it's a problem. There are rootstocks that do show it. I'll give you an example, Dusa that has phytophthora root rot tolerance, and it also has some salinity. But nobody's growing it here,
So,
for clonal.
Clonal rootstock production may be a good opportunity for somebody here. in
It could be an
Florida.
opportunity.
Maybe outside of Dade County, a little further north, it might be easier.
No, I mean, I don't know. It's just a matter of learning how to do, perfecting the thing. Bruce mentioned that there are people now doing tissue culture, producing avocados. I mean, that's another way to do it potential, but there's nobody doing it. And, when you use clonal rootstocks or tissue culture plants, as Edwin mentioned, it's a lot harder to get the trees established. The trees tend to be weaker, they don't establish as quickly as if they're on a seed rootstock. So, yeah.
So are you guys saying, the phytophthora root rot and the laurel wilt, like pretty much ubiquitous, like across all farms that are growing avocados in Miami-Dade County? Like, is there anyone who is escaping these issues?
If you're outside of the 12 states that have laurel wilt,
Yeah.
you're escaping it for now. The thing of it is these ambrosia beetles, as Romina alluded to, they're moving through the natural areas and urban areas, attacking trees in Lauraceae or avocado trees in dooryards. And they're moving west and northwest. They're already into Texas. And I think up to Tennessee, North Carolina. And so they are moving slowly. I remember seeing a number when this problem first arose. The red bay ambrosia beetle, which was the original vector of this disease, the original beetle, they estimated would take over or move 25 to 30 miles in a year. So it's moving slowly. So we figured, there's some natural barriers between Texas and Mexico and Texas and California, but we also found that people moving trees or germplasm or infested wood like firewood
Yeah.
would move the disease hundreds of miles. Only takes one introduction.
Yeah. So you're not saying anyone, escaping these, issues like via like cultural practices, like within the orchards?
Yes, no, cultural practices, yes. So that's why we're talking about, putting trees on beds,
Yeah.
having well-drained soil, pruning, if you have mature trees, regularly pruning your trees to open them up to light and to air movement to try to tamp down the beetle activity. So there are some cultural practices you can do. It's just that we don't have something that's like, take this aspirin and you're good to go, call me in the morning. It's not as simple.
Well, is it as simple as like, I guess the farmers who are experiencing the issues, like adopting those cultural practices, or they don't like, I guess I'm just trying to understand, do they not want to adopt those cultural practices?
No, people have.
But they're still having issues.
Yes, it's not 100%.
Okay.
Yeah. So when I said, like with the proof, it tamps down the issue. Tamps
Right.
down, it does not eliminate it.
Yeah,
So, and people have been adopting this.
yeah. Can it get it down to a level that is acceptable, or is it still gonna be, economically unviable for people?
Well, it's not that it's not viable. It has more, the viability of the avocados has more to, right now, seems to have more to do with the competition from offshore and that we have low production because
Yeah.
we don't have enough for the pack out. Go ahead, Carlos.
To answer the young lady, I'm a grower.
Yeah.
I do different cultural practices,
Mhm.
maybe in a unique way,
Yeah,
but my growth for the last 2 years, even with the wilt, even with the phytophthora, I'm turning a profit.
yeah,
And I'm not complaining, if I could keep it this way, it will continue to be profitable, even with the competition from overseas, just based on my cultural practices, right?
Yeah,
So it is possible.
so are you losing like significant number of trees a year, or are you kind of holding
I
steady?
went from like losing, I keep a moving average,
Yeah,
so like 2 years ago, I had a moving average of about 10 trees a month.
yeah,
Now down to like 0.2, 0.3.
so better survival,
Oh, yeah, the moving average is going down.
yeah, so did you change something that?
Oh,
caused
yeah,
that?
it's a lot. I mean, the pruning, first thing, the pruning and topping. I do allow, the moment I finish harvest, pruning and topping.
Mhm,
I keep my trees very healthy.
mhm.
How do I deal with the phytophthora issue and the problems with the dieback? I do a lot of, I apply a lot of trichoderma,
Yeah,
I apply a lot of Bacillus subtilis,
mhm,
and they keep the trees clean and happy. So happy trees, less beetles.
yeah,
So I saw a dramatic drop from 2 years ago to now.
But you also do, very good surveillance.
Oh, yeah,
So
I'm there.
monitoring the trees and extracting the symptomatic tree right away.
I'm probably the only guy that just takes the whole tree
Yeah,
out with
you-
roots
you
and
kind
all.
of light off the.
Huge machine comes and takes the whole tree out with roots and all. I
Yeah.
take it to what I call my avocado cemetery where it gets grown. But my avocado cemetery,
And how do you treat your avocado cemetery?
2 years ago, it was, dozens of trees.
Do you burn them
Now,
or?
it's just, weeds are growing.
Are you shredding those?
Yeah,
Are
we
you
are.
shredding those, kind of, what do you do?
Yeah.
The cemetery, do you burn
Yeah,
it,
in
you shred it?
theory, yeah, I spread them in place in an area.
Yeah, yeah. So,
Believe it or not, none of the adjacent trees have been
Yeah,
contaminated.
no,
Yeah,
it's fair.
so, yeah. So another cultural practice, so for instance, when we first started recommending people remove trees, you were looking at $150 to remove a tree, and it was very laborious because the traditional way is you would chainsaw the tree and then put it through a chipping machine. And then you would take a backhoe and dig the stump out, set it down, and then try to light it on fire. And it would take days
Yeah.
and days and days.
Good thing, every time you do that, I will lose like 5 pounds. I get home to my scale, 5.
So now we have the machinery and the know-how where you basically knock the tree over, drag it out, run over it with these shredders, and the tree's gone in about 15 minutes.
okay. So that makes
And the beetles really can't survive in the chips. And the disease doesn't survive in the soil, doesn't, that kind
Yeah.
of thing. Yes.
Question online for you, Carlos. What variety are you growing?
How
I
many?
am growing Donnie's, Monroe's, 7515s and, betas. And I have like probably 50 or 60 varieties of,
Okay.
which I call my experimental varieties, growing.
Got it. Thank you. Jonathan, do you have the QR code for the survey?
Yeah, sorry.
So the online people can.
How many acres?
Yeah, so if you would,
Okay.
well, whether you're online or you're here.
Yeah,
We have a survey, of course, we always wanna know, I loved it, hated it, it was worth coming, it wasn't worth coming. Don't ever contact us again. Look at the QR, yeah, if you could, do the QR code and do the survey. That would be helpful for us, yes.
Has anybody looked at increasing the tree planting space so that the roots aren't? Or is it not,
Yes.
it's just not viable, I mean, it's not,
Well, yeah, I mean, you'd have, it just depends on the age, not just, the age of the tree and how closely they're spaced, right? So the more closely they're spaced, the more likely they're to, in a shorter period of time, attach and root graft. Whereas if you're, so I wouldn't recommend, in fact, you bring up a good point. We did see some people replanting their avocados at like a 10-foot spacing. Because I totally get it. They want to get back in production. They want to have trees right away. So let's double up on the trees. The problem is they're creating, shaded conditions. Root grafting is going to happen much sooner. So I would recommend it's better to just go back to your 20 to 25-foot spacing as you had before. Because getting them too crowded, you're going to end up, and people say, well, in 10 years, I'll take every other tree out. Well, that never happened. I mean, I see some groves where they were planted in the 70s and they were planted when high density was like a fad back in the 70s and people were planting at 10 feet. And that grove has had nothing but nightmare with laurel wilt for the past, 10 years.
I'd like to comment on that whole thing. The way you plant a grove will make a huge difference, but two things I want to say. We cross-trenched when we put our grove in. That helped us with hurricane resistance, as long as we keep the trees lower than 12 to 15 feet tall. When a hurricane comes, they don't pop out of the ground. You may break roots, branches, and if you're higher, you may crack the tree, but that does not pop out of the ground. You make holes, round holes, and one of the problems with round holes is as the drill is going, it's sealing the edges. And it's like the tree went into a pot.
So you plan on the cross, is what you're saying?
We planted on the crosses, and I'm gonna get to it. When you drill holes, the roots tend to stay in there, because the other thing you tend to do is you tend to not put what you dug back out, you try to amend the soil, put nice soil, no. Just put right back the rock that came out. Then the tree is forced to try to find its way out, but it's harder because we have our rock, limestone rock is what concrete is made out of, and that's
Yeah.
made with concrete wall and everything, with just from the heat of the abrasion of the drill. So in my view, it's better to do something like a cross trench. However, where we had the problem is the roots follow the path of least resistance. So where are the roots gonna go?
Right now.
In a square, in a cross, and they're gonna go right to the next tree, and so that root grafting will occur. We planted at 20-foot centers, but still, that's how the laurel wilt started to spread straight down roots. If you don't have things like irrigation lines running underneath there, you're great, because then what your management can do is you just
Retrench.
just trench across between those rows to keep those roots from grafting, or to break that ground. If you catch that early enough, then you can keep that up as a management practice. Your trees will be more hurricane resistant. They still will have the opportunity to have their roots a little deeper in the ground. Our trenches were originally nominally 24 inches. They were more like 18 by the time to soil.
So how many years in between trenching would you have?
Well, that's the thing we have to look at. I don't know that
Nobody
anybody's
knows.
really done this.
The main thing is, we had people try the trenching. We said, why don't we just go ahead and settle the roots, so you'd have a check report.
Yeah.
First of all, you're gonna wanna wear ankle braces when you walk through the grove, because you're gonna fall in those trenches.
But you don't have to
But-
trench all the way across each
Yeah.
row, just really across where that basic
But the
tree,
worst,
the drip line.
the worst part was what you mentioned though, is people have underground irrigation there.
Yeah, and that's the
And
thing,
so
but if you
now I have
plan
to
your
agree.
road, you're planting ahead of time, if you're in control of where you're setting things, don't make that mistake of putting your irrigation in there with it. Put it in between or on the side to where you're not gonna kill it.
Somewhere.
One more question. So I don't think it really got touched on. It seems like the symptoms for the phytophthora root rot and the laurel wilt appear very similar. Is there, how do you know which one you have, so you don't just go rip out your whole tree?
Well, a couple things. One is, did we just have a flood?
Yeah,
If we had a flood, you're probably seeing the results of phytophthora. But the other thing is you can test, so you can take wood samples or you can take root samples, and we have a plant clinic, Romina runs a plant clinic, and you can determine.
But if phytophthora
The other
becomes
thing-
ubiquitous, it would, difference is having a soil test.
But
That's fine.
if you want to know whether it's phytophthora or laurel, it could,
It could be both.
both, actually.
Yeah.
Yeah, so
The best way I would say is to follow the symptomatic branch, not completely dry, because if you do dry, the vascular system is gonna be discolored because of that.
yeah,
But to follow the early wilting
yeah,
and then open, it's really easy to take out the bark of the avocado.
yeah.
And if you see streaking or you see discoloration,
Yeah.
it's most likely laurel.
Yeah, like a darker coloration.
Correct.
Yeah.
Like what color, like coffee?
well, it looks like streaks, brown and
So
black.
the practice
streaks.
would be the same. You basically wanna trim off that dying thing.
You want to take out.
But I'm saying, you can make your
All
first cut and look, you got laurel wilt, okay, you got it.
Yeah.
If you don't have it, then you can not trim the rest assuming it maybe just be phytophthora root rot and